Today we're going to be discussing research done to explore why, while sexually experienced males copulate more often than sexually inexperienced males, there still lies a great variability between their behaviors. It is known that glutamate within the medial preoptic area of the hypothalamus plays a role in sexual behavior. This is known because glutamate levels within the MPOA rise and fall during sexual activity, peaking with ejaculation and falling off during the PEI, the post-ejaculation interval. When rats were given glutamate uptake inhibitors, the ejaculation latency and post-ejaculation interval decreased significantly. You may be wondering what ejaculation latency and post-ejaculation interval even mean. In the context of this experiment, ejaculation latency can be defined as the amount of time before each climax, and post-ejaculatory interval is the amount of time needed for recovery between the climaxes. Decreasing ejaculation latency and shortening the PEI means males can climax faster and have more than one orgasm in a sexual encounter. Researchers wanted to know if differences in the number of astrocytes, which are the cells in the medial preoptic area that produce glutamate, could account for the differences in sexual behavior between experienced and inexperienced males, and if the number of astrocytes are related to ejaculation latency and the duration of the post-ejaculatory interval. An experiment was designed where 30 male rats were randomly divided into two groups of 15. One group was considered the sexually experienced group. To achieve sexual experience, they were given 6 controlled 90-minute sexual encounters with a hormonally primed novel female. During the 6 experience, males were tested for sexual proficiency, meaning they needed to achieve 2 ejaculations and initiate a third mating bout in a 90-minute interval. Both groups then underwent a sexual behavior test where mount, intermissions, latency to ejaculate, and the length of post-ejaculation interval were scored. Subjects were then sacrificed for immunohistochemistry. During immunohistochemistry, the brains were sliced and the astrocytes present in the MPOA were counted. A two-way analysis of variance and Pearson's correlations were performed to compare the number of astrocytes with ejaculation latency and the duration of the PEI. The number of astrocytes present in the MPOA did not differ between the two groups. Results show that compared to experienced males had larger ejaculation latencies. The duration of the PEI did not differ between the two groups, and the number of astrocytes did not vary with length of the PEI in either group. In previous studies, lesions to the MPOA in sexually inexperienced male rats caused the elimination of sexual behavior, but lesions to the MPOA in sexually experienced rats does not cause change in sexual behavior. This may be because that with continued sexual experience, glutamate in other parts of the brain, like the medial amygdala, becomes the dominant modulator instead of glutamate in the MPOA. This would explain the discrepancy in our results, where the number of astrocytes in the MPOA correlates with latency to ejaculate in sexually inexperienced males, but not in sexually experienced males. In conclusion, the hypothesis that differences in number of astrocytes in the MPOA accounts for sexual behavior variability between experienced and inexperienced males was rejected. Although a rejected hypothesis is not a flaw within itself, the experiment possessed flaws that may have led to a rejected hypothesis. There were errors in method that caused the loss of brain tissue during slicing for immunohistochemistry examination. Some of the rats did not meet the criteria for sexual behavior during the test to be included in the data. This left experimental groups uneven in number. Considering the desire to have experimental groups as identical as possible, this may have caused issues for data analysis. The authors also chose to operationalize astrocytes as having 3 or more GFAP positive processes, converging on the GFAP positive cell body. The authors admit that not all astrocytes express this behavior and in turn may have caused them to underestimate the total number of astrocytes present. Results may have differed if they operationalize an astrocyte under different criteria. Despite a rejected hypothesis, multiple possible explanations for the results were provided in the discussion, which demonstrates the possibility of further research. The researchers could have then included a possible experiment design as a next step for future research. Overall, the experiment was executed in a proper manner and results discussed appropriately, making for a significant contribution to the scientific community. Remember kids, it's okay to try and fail again, but it's not okay to try and fail to try again.